Hi, and welcome to my presentation on microbial growth and its control. So first we'll talk about microbial growth, then its control. For normal organisms, when we think about growth, we think about the organism getting bigger or maturing. But for microbes, it's different since they're just a single cell. They don't really get bigger, they just divide and make more of them. So for microbes, the definition of growth is an increase in the number of cells present in the population. So the microbiology definition of growth is an increase in population size or increase in number of cells. There's different ways that bacteria can divide and different ways that other microbes will reproduce. Almost all microbes will reproduce asexually or at least have the ability to reproduce asexually. But the most common method of cell division for bacteria is called binary fission. In binary fission, the cell basically will grow until it's basically twice the size that it originally was, and then you have a structure form in the middle, which is called the septum, that splits the cell into two, and then you end up with two different cells. So in the diagram here, you have a typical bacterial cell that first grows to double its size, and then you have the septum form in the middle, and that septum will divide the cell into half and split it into two daughter cells. So this is the process of binary fission. We describe microbial growth using the term generation time. The generation time is how long it takes for microbes to divide, usually by binary fission, but by whatever mechanism they're using to divide. Different microbes will have different generation times. The shorter the generation time is, the faster that type of microbe will grow. Usually for bacteria, the generation time would be measured in hours or days, but there's some exceptions. For instance, the bacteria that causes tuberculosis has a very long generation time and takes many days to divide. Microbes will always be growing exponentially. Even if they have a slow generation time, their growth is still exponential. Their numbers will double every time, every generation. So however long the generation time is, that's how long it takes for the number of cells in the population to double. Anytime you have the population doubling on any time scale, that is exponential growth. So when you start out with one cell, within one generation time, you'll have two cells, then 4, then 8, and 16, 32, 64, 128, and it starts getting faster and faster from there, and the numbers grow larger and larger. In order to graph microbial growth, we will frequently use a logarithmic graph, where you have the time plotted in hours or minutes or days, depending on the generation time, and then the number of cells on the y-axis will be going up by the log, so that you can get a straight line. There's two basic ways for microbes to grow and to live: planktonic and sessile. In planktonic growth or planktonic lifestyle, microbes are living basically in a liquid like water, but that could also, well, yeah, water, usually water. Could be another type of liquid like oil, but usually that would be water. So that would mean microbes are suspended in the water or swimming through the water, but they're not attached to anything. In sessile growth, the microbes are living attached to a surface of some kind. So they might still be underwater, but they're attached to something like a rock or something. It's only the sessile cells that are going to form a biofilm, and you can have a lot of microbes that will do both types of growth that kind of switch between them. So typically you'll have planktonically growing cells swimming around in a liquid, and they will happen across a surface and attach to it using their fimbriae or their hemi or their pili. And once they're attached to that surface, their glycocalyx will start forming the biofilm if they have a slime layer glycocalyx. And then they'll basically switch to a sessile lifestyle and grow inside the biofilm. And the biofilm will be protecting them from toxic chemicals in the environment. But not all sessile cells will form a biofilm, just many of them will. When you see algae growing on rocks, That is a biofilm. That's a type of sessile growth. That's a biofilm. So now we'll talk some about how we grow microbes in the lab. There's two basic types of culture that we can use for microbes, batch culture and continuous culture. We'll talk about continuous culture in a moment. First, batch culture is probably the most common type of culture because it's cheaper, but it's also requires more care of the cells. You have to take a more active role in caring for the cells. So in batch culture, you're growing the cells in some type of container. Could be a flask, any container really. So they're basically grown in batches that are contained in these containers. Anytime you're growing microbes in batch culture, there's 4 phases of growth that you can observe. First you have the lag phase, then exponential phase, then stationary phase, and finally death phase. In the lag phase, you have basically added the microbes to this container with their media, but they don't grow yet. So their population numbers are remaining constant when they're first added. And that's basically because they're making things that are required for cell division, so they haven't divided yet. If you are using batch culture, then you're going to need to periodically change your containers, or at least change the media, depending on how you're doing it, because they're going to use up all the nutrients and their waste products will build up in there. So they need it to be changed periodically. If you're changing to a new type of media, then you're going to have another lag phase. If you're changing the media and replacing it with the same type of media, you generally would not have another lag phase after you've changed the media. And the same for the container. If you're changing them to a new container that's in the same conditions, with the same media and the same basic conditions, then you may not have a lag phase observed. They may go straight to the exponential phase. In the exponential phase, your population is basically growing as it normally would, basically doubling according to the generation time, so growing exponentially. During this phase, the cells are kind of the most active and the most healthy. You have a lot of nutrients available in the media. There's not too many waste products in the media yet. So the cells are generally doing very well. Because of that, we usually like to do experiments on cells that are growing in the exponential phase. So you have to kind of time your experiments to work around the exponential phase in your batch culture. Once you have run out of nutrients, you're going to leave the exponential phase and the cells will not be as active, and that's usually not a great time to be doing your experiment. After the exponential phase, you enter the stationary phase, in which the population of cells remains basically constant, which means that the cells are dividing, but they're also dying. So as new cells grow, you have old cells die, and the population is basically remaining stable. In this phase, the nutrients in the media have been depleted somewhat, so there's not as much nutrient availability, and that's why the cells are not dividing as quickly as they were before, so that the number of cells that are dying is able to kind of balance out the number of cells that are being sort of born by cell division. Another thing that happens in the stationary phase is that the waste products of these cells are accumulating in the media, and that can kind of inhibit the growth of new cells. The cells will overall be less active. Their metabolism will slow down because there's less nutrients and there's more waste products around. After the stationary phase, as nutrients continue to decrease in the media and waste products continue to accumulate, you will enter the death phase, in which the population is actually declining. It actually will decline exponentially, but not according to the generation time. It'll be a much slower decrease than the generation time increase in the exponential phase. And it's basically just occurring because there's not enough nutrients to sustain cell growth or even cell life. And the waste products are quite concentrated in the media, and they become toxic when they're at high concentrations. So generally you don't want your culture to enter the death phase. You would want to replace that media before that happens, so that you're getting rid of waste products and delivering more nutrients to the cells. In the graph here, we have first the lag phase. When you add the cells to this culture, Initially, they do not grow, and then when they start growing, once they've synthesized everything they need for cell division, they start growing. They'll grow exponentially based on their generation time. So that's the exponential phase. Then you'll see the population numbers level off and remain constant, which is the stationary phase. So you have less nutrients available in the media now and more waste products, so they're dying more and growing less. Death is equaling growth. And then you transition into the death phase where the population numbers begin to decline because you have not enough nutrients left and too many waste products to sustain the life of those cells. If you're working in continuous culture, that means that you have a system set up where fresh media is always being added to the culture while the old media is being removed. So this is a more complicated setup, but it requires less work out of you. If you're growing cells in batch culture, that means that sometimes you need to come into the lab late at night or on the weekend or something to replace some media for your cells. If you're working with continuous culture, then you don't need to do that because the system is doing it for you. A nice thing about continuous culture is that you can have the exponential phase of cell growth last for a very long time. Basically, the cells should always be growing in the exponential phase as long as the system is set up correctly, which means that you can have an experiment running on those cells for a long period of time. As you're always adding new media and taking old media away. So you're always adding new nutrients and taking away the waste products so the cells can remain healthy and active and growing exponentially. There's different systems that you can use for continuous culture. The most common one is called the chemostat. And when you're setting up your continuous culture system, you're looking to reach the steady state. In the steady state, you actually have no change in the population of microbes. So normally in batch culture, in the exponential phase, you have an exponential increase in the population of microbes. In continuous culture, when you're at the steady state, the desired state, you don't have a change, even though the cells are growing exponentially still. The reason is that you're also taking away cells when you take out the old media. There's no way for you to remove the media without removing cells too, unless you're going to filter it, but you don't really want to do that. It's actually better if you do remove some cells with the old media. So the rate at which you're removing cells equals the rate at which you're adding cells by the generation time, by cell growth, according to the generation time. Therefore, the population of microbes will remain constant. And here's a basic diagram of a continuous culture system of a chemostat. So you're adding new media constantly, and you're always stirring it so that the concentration of nutrients and waste products is even throughout the container where the microbes are growing. And you also have media being removed constantly. So this media that's being removed is going to have waste products. It'll also have some nutrients in it, of course, and it'll have cells in it. But you're always adding in new media that has new nutrients so that the media inside of this container always has a high level of nutrients and a low level of waste products, which is really nice for the cells. There's different types of media that you can use. In general, all media can be classified as either solid or liquid. Solid means usually you put agar in it. There's different ways to make a solid media, but usually means you added agar, which is derived from the cell walls of algae, actually. Liquid media is just, a liquid. Apart from that, the components of the media would be pretty similar between solid or liquid media. So in general, to make a solid media, you can just take your liquid media and add agar to it. If you're growing cells in like a petri dish, that means you want solid media. For most purposes, you're going to use liquid media. Like if you're using a flask or something or a continuous culture system, that needs liquid media. Then all media can be classed as either defined or complex. In defined media, you know exactly what's in it and you know the concentration of every component of that media. That means that you had to make it yourself or you bought it pre-made from a company. So it's basically artificial or synthetic media. In complex media, you don't know exactly what's in it and you don't know the exact concentration of the different components. So that means that you could also have, bought it from a company, of course, but that means that it has something in it that is coming from a natural source. Could be from cows or from yeast. Those would be common sources for complex media. So you're basically taking some natural extract. You don't know exactly what's in that extract. It just has a bunch of stuff that you know is good overall for the microbes, and you're adding it to your media. The benefit of a complex media is that it's simpler to make. So microbes will need a lot of different growth factors in small amounts, different vitamins and such. If you're making a defined media, you have to add every single one of those by hand, and it's quite laborious. In a complex media, you're just taking this natural extract and figuring that it's all in there. You're taking like some cow's blood or something, you're figuring all the vitamins that these cells need, they're inside of there, so it's fine. So complex media is easier to work with or easier to produce because of that, but you do need to bear in mind that you don't know exactly what all is in it. There's also a special type of complex media called enriched media. In enriched media, you're using some really nutrient-dense natural extract to add a lot of growth factors to the media. That would usually be blood or serum. Serum is just blood with the clotting proteins removed from it. So a lot of the time you'll see fetal bovine serum or FBS added to media, which would be making that media enriched. So microbes that have a really high requirement for growth factors, they really need a lot of different growth factors. You're usually going to supply that using enriched media that has FBS or some other blood product in it. Those microbes that need a lot of different growth factors have a special name. They're called fastidious microbes. They can be very hard to culture in defined media since they need so many different things. You'll be there forever adding every little thing that they need into that media. It's much easier to use an enriched media where you just added some FBS to it and you're good to go. Then you can also have special medias that are selective or differential. In a selective media, you are either putting something in that inhibits the growth of certain microbes, or favors the growth of other microbes, or you're leaving something out that favors the growth of some microbes over others. So it's basically media where not all microbes will be expected to grow equally well. You're expecting certain microbes to grow well on it and other microbes to grow poorly on it. So you might change the pH of the media so that only microbes that tolerate a low pH will be favored to grow. Everybody else will have a hard time growing in the low pH of the media. You could add extra salt to the media. You could restrict the nutrient sources in the media. So if you leave out glucose or some other sugar from the media, then any microbe that really needs glucose to grow, it's not going to be able to grow well. Microbes that are able to do without glucose will be able to grow well. The point of your selective media is that you're favoring some microbes over others. So if there's a specific type of microbe that you're trying to grow and you're not trying to grow others, but you have kind of a mixed sample or a heterogeneous sample, it's not a pure sample of one type of microbe, but it has lots of different microbes in it, then you might use a selective media to kind of weed out the microbes that you don't want to grow and favor the microbes that you do want to grow. Enrichment media that we had mentioned back in chapter 1 is actually a special type of selective media. And it's a type of selective media where the specific purpose of the media is to favor the growth of microbes that are rare in your sample or that grow very slowly. For instance, Mycobacterium tuberculosis that causes tuberculosis grows very slowly. So if you have a heterogeneous sample with a lot of different microbes in it, and the one that you're interested in either grows real slow, or there's just only a few examples, a few cells of it in that sample that you have, it's going to be hard for you to find that microbe and isolate that microbe so you can work with it. If you use an enrichment media, it's going to be much easier to help that microbe grow while inhibiting the growth of other microbes so that they don't crowd out the microbe that you're actually interested in. Then once you've got that microbe that you are interested in grown up to where you have like a significant population of it, then you'll probably be able to take a sample of just that microbe and grow it in pure culture with just it, and then you can actually study it. Finally, you have differential medias. Differential media is basically media that's telling you something about what's growing in it. So it'll have some type of indicator that usually changes color depending on what's happening. And usually it's related to the metabolism of the microbes growing on it. So you'll have you'll have media that changes its color if the microbes are doing fermentation of glucose, for instance, or if they're doing fermentation of sucrose. Or depending on the pH of the media. So if they're producing some acidic waste product, then the color of the media will change. Just something that tells you something about what those microbes are doing. They're growing in the media. That lets you see really quick at a glance what's happening, and it can help you ID the microbes without doing more complicated tests on them, other than just growing them with this special media. Whenever we're growing cells in culture, we usually have a need to count them, and there's different ways that you can do that. So first you could do a total cell count, where you're basically just trying to count every cell that is in your media, that's in your sample or that's in your culture. So you're actually going to take a sample out of your culture, a small volume of the media with the cells in it. You'll take it out of the culture container and add it to a grid, basically a glass plate that has a grid on it, and you'll look at that under the microscope and you'll count the microbes or you'll count the number of cells that are within the squares of the grid. And that can be kind of hard to do because you have to keep track of which cells you counted and which ones you didn't, and of course which number you're on. And then once you have that count, you'll do a calculation to get the number of cells per mil in your original culture vessel. So that's a good way to count cells, but it's kind of hard because it could be hard to keep track of how many cells you've counted if there's a lot in your sample. Also, it doesn't let you distinguish between living cells and dead cells. If you just see these cells in the microscope, you can't tell if they're alive or dead. So you're going to count the dead cells too. Unless, of course, you've stained them in some way. So there's different stains that you can add that would tell you, like if the cell is alive or dead, that'll stain it a special color if it's dead, and then you can just not count that cell. Or there's even stains that will tell you like which species you're looking at. So if you have a heterogeneous sample and you're only interested in the cell count for a specific species, you could add a stain that colors that species a color, and then you can count only the cells that have that color. So stains for your total cell count can make it more useful for you. You can also do a viable count. That's also called a plate count, or a viable cell count, or a plate count. And that type of count, you are going to plate your sample. You're going to take your sample from your culture vessel and plate it onto a petri dish and grow it there. And you're going to get colonies, and you're just going to count how many colonies you have. So what's good about this method is that you're only counting viable cells, because if you have a cell in your sample and you put it on the petri dish and it's dead, it is not going to make a colony. Only the cells that are alive will make a colony. It can be a kind of a difficult technique sometimes because you're counting colonies, not actual cells. So you have to kind of make sure that when you plate those cells onto the petri dish, you spread it out good. That way the cells are all separated from each other and each cell will give rise to one colony, and you don't have just colonies running into each other and making a big mess that's impossible for you to count. Also, when you spread these cells out, you don't actually know if they're one cell that gives rise to a colony or a little clump of cells that gives rise to a colony, which means that your count is not going to be totally accurate. And because of that, when we do a viable cell count or a plate count, we're not counting ultimately the number of cells, we're counting the number of colony forming units, or CFUs. So a colony forming unit ideally is just one cell that then gives rise to a colony, but it might be a clump of cells that gives rise to a colony. So a colony forming unit takes into account the fact that you don't know if you're starting with a single cell for each colony or a little clump. A good thing about doing a plate count is that you could use selective media so that only the microbes that you're actually interested in counting will be growing well. Or you could use a differential media as well to know something about those cells at the same time as you're counting them. You can also count cells using what's called turbidity, or the cloudiness of a media produced by cells. So when you have medium, it's usually clear and you can just see straight through it. Once the cells are in there and they're growing and they reach a high concentration, the media will get cloudy. So you can't see like individual cells, of course, because they're microscopic, but you can tell that the media is cloudy because of how many cells are in it. And you can actually use this machine, the spectrophotometer, to measure exactly how cloudy the media is. So you'll get a number called the optical density that indicates how much light is able to pass through the media. The more cells you have in the media, the less light is going to be able to pass through, because light will be bouncing off of the cells instead of just going straight through the media. So over a certain range of optical density values, you can relate that optical density to the turbidity or cloudiness of the media, and then to the amount of cells that are in the media. So it's only accurate over a certain range. Once you get so many microbes growing in that media that no light gets through, you could add more microbes and you wouldn't be able to tell. You're still going to have the same amount of light getting through, which is none. So you have to be careful that the optical density value you're looking at is within the range that can be accurately used to estimate the cell number or the cell count. Okay, so now we'll talk about some of the environmental variables that impact microbial growth. And the main ones would be temperature, pH, oxygen presence, and water. With water, we mean both how much water is there, and we mean also how many solutes are dissolved in that water, which is the osmolarity. So microbes will be affected by the physical presence of water, and they'll also be affected by the amount of solutes within the water, or the osmolarity of the water. So we'll go through each of these variables in turn. So temperature is the average movement of molecules in a substance, or the average kinetic energy of the actual molecules. And kinetic energy is energy that's associated with movement, so temperature is just the average of the movement of the molecules in a sample. Temperature affects microbes very significantly. When the temperature goes up, that generally means that chemical reactions will go faster. So that's usually a good thing for microbes. When their reactions go faster, they grow faster. But when you get to a certain point, high temperatures will begin to denature the proteins or cause the proteins to unfold. And when proteins are unfolding, they're losing their function. If you lose the function of your critical proteins, you're going to die as a microbial cell, or as in any organism, really. And this is the reason why high temperature is dangerous for organisms and also for microbes, including for microbes. In addition, if the temperature is really high, it permeabilizes cell membranes, which means that it makes it so that the membrane is really easy for things to get through. So that might mean that you have kind of toxic products coming into the cell that should have been kept out, but that also means that all the glucose and solutes that need to be inside of the cell, all the stuff the cell is trying to hold inside of itself, it can't do that anymore, because now those things are just going to leave through this permeabilized membrane. A permeabilized membrane is not an effective barrier between the cell and its environment. So if a cell loses that barrier, it basically will die. So that's how high temperatures kill cells. Low temperatures are also bad for cells. They, of course, will make reactions slow down. So microbes will grow more slowly at a low temperature, but they'll also make membranes very stiff if the temperature gets too low. And if the membrane is very stiff, then it's hard for the cell to transport things across it. So it can't get nutrients to come into the cell through the membrane, and it can't export waste products out of the cell across that membrane either. And that means that the cell will basically be starved for nutrients and have waste products accumulating in it, and it'll die. So every microbe has a different temperature tolerance, is able to tolerate a different range of temperatures. For any given microbe, the range of temperatures that it prefers, or the range of temperatures that it tolerates, corresponds to the temperatures that are found in its natural environment. So microbes are basically adapted to the conditions where they would naturally be living, which makes sense. For any microbe, we can identify 3 cardinal temperatures, which are the minimum temperature that a microbe is able to grow at, the optimum temperature that a microbe can grow at the best, and the maximum temperature that a microbe could grow at. So on this graph, you have the minimum cardinal temperature. Below this temperature, this microbe will not be able to grow. It will not be able to reproduce. As the temperature increases, you're going to have your reaction speed increasing, so the microbe is going to grow faster and faster until it gets to the optimum cardinal temperature, the temperature at which it grows best. And then shortly after that, shortly past that, you hit the maximum temperature. Beyond that temperature, the microbe is not able to grow. The optimum temperature is generally pretty close to the maximum because as temperature goes up, reaction speed will go up. So microbes will go faster and faster. Once you go too far, though, then your really critical proteins will begin to denature and you hit the maximum. So the optimum is generally going to be like just below the maximum. It's how much temperature you can tolerate, so how fast you can get your reactions going without denaturing those proteins that you have to survive. We can group microbes based on their tolerance for different temperatures, or based on the range of their cardinal temperatures. And there's 4 major groups of microbes classified in this way. We have the psychrophiles that like it cold, the mesophiles that like it medium temperature, kind of the temperature that we would like it at, thermophiles, microbes that like it hot, and hyperthermophiles, the microbes that like it really hot. Mesophiles are usually the most relevant microbes for us in terms of our health. Any microbe that is going to be living inside of you or inside of some other mammal is going to be a mesophile. It's needing to tolerate temperatures that are within your body, which would be mesophile type temperatures. A mesophile will usually be happy at around 70 degrees Fahrenheit to 105 degrees Fahrenheit. So that's basically between room temperature and your body temperature if you have a fever. That means since their optimum temperature is going to be just below their maximum, that means that their optimum is going to be pretty similar to your actual body temperature when you're healthy. So that goes for the microbes that are part of your microbiome, not the ones that live on the surface of your body like on the skin, but the ones that live inside of you like in your gut. They are mesophiles. They like it to be at your body temperature. Although skin microbes would also be usually mesophiles, your skin is more likely to be at like room temperature. So it wouldn't be quite at their optimum, but they would be fine for them still. Pathogens, any pathogen that's going to cause disease in a human, it has to be able to survive in the human body, which means that it needs to be a mesophile for the most part. So almost all human pathogens are mesophiles. There's a few exceptions to that. But any microbe that's going to be successful in the human body needs to at least be able to tolerate your body temperature. Most of them will grow well at your body temperature. So their optimum temperature is like your body temperature. Because of this, mesophiles are also the best studied microbes. Well, I guess partly because those are the ones that are most relevant for your health, but also because those are the ones that it's easiest to culture because you don't have to make it super hot or super cold for them to be happy. So we'll look more at the psychrophiles and then at the thermophiles since they're kind of special. A psychrophile is some microbe that needs it to be cold or likes cold temperatures. A true psychrophile will need it to be cold all the time. They cannot handle a hot or a warm temperature. So they're going to need to live in some environment that is not only cold in the winter, but it's cold in the summer too. That means that most of them are going to be living in the ocean, like down deep in the ocean where it's always cold, or near the poles, the Arctic or the Antarctic. It's pretty hard to culture psychrophiles because they cannot tolerate temperatures that are not cold. So if it's just heating up to room temperature, for a psychrophile that's really hot, it's already going to be dying at that temperature. So that just makes it really difficult. You can never expose the psychrophile to the temperatures that are kind of in the room that you're working in. So it's difficult to culture them. They will also grow slowly. Since reactions proceed slowly at cold temperatures, their reactions basically are just designed to go real slow, and they're always going to grow pretty slow. You have also another group of microbes called the psychrotolerant microbes that actually like mesophile temperatures, so kind of those room temperature to body temperature temperatures, but they are able to still survive and even grow a little bit at the low temperatures. So a psychrotolerant microbe is not a true psychrophile because it does not need consistently cold temperatures. It would actually prefer if the temperatures were not so cold, but it can still get by if the temperature does drop. So there's a couple of human pathogens that are actually psychrotolerants. The most important one historically is Yersinia pestis, which is the bacteria that causes the Black Death or the bubonic plague. So that microbe is able to still survive through the winter, and do okay at cold temperatures. You also have Listeria monocytogenes as a microbe that is able to survive at low temperatures and kind of grow a little bit, and it can cause disease. Mostly it only causes dangerous illness for pregnant women. So if you've ever been told or heard that a pregnant woman shouldn't be eating like ham or deli meats like that, it's because those types of meats can have Listeria monocytogenes on them, and that can survive in your refrigerator. And then you eat it, and it will cause serious disease potentially if you're pregnant. If you've ever seen snow that has a color to it, that's probably because there's microbes, psychrophile microbes growing in that snow, or maybe psychrotolerant microbes as well. So in this image here, all this red kind of dusting on the snow is actually bacteria that are growing, psychrophile bacteria that are growing in the snow. It can be other colors as well. It could be yellow. So the yellow snow is probably got bacteria in it. It's not urine, it's bacteria. Psychrophiles have a lot of special mechanisms that they use to grow at these cold temperatures. Their enzymes are not the same as enzymes in a mesophile or a thermophile. They're more flexible than a normal enzyme. So since the enzyme of a psychrophile is more flexible than normal, that means that it's easier for it to change its shape and it's easier for it to unfold. So if the temperature gets too high, what would be a normal temperature for you and me, for a psychrophile enzyme, that temperature is going to denature it. It's going to unfold. But it's going to maintain its flexibility at a cold temperature. The majority of enzymes need to actually be able to change their shape a little bit in order to have their function. That's called a conformational change. If they can't change their shape at all, then they're not going to function. So a normal enzyme, like from you or me, that we put at a very cold temperature, it's not going to be flexible enough to undergo a conformational change or a conformational shift at that low temperature. It's going to be too solid, too gelled up kind of. So it's not going to function. A psychrophile's enzyme that is more flexible than normal will be able to still change its shape and function at a cold temperature. Their membranes are also specially designed for the cold. They are more fluid than a normal membrane like you or I would have. So even though membranes get stiffer at cold temperatures, these psychrophile membranes are actually reaching their ideal fluidity at a cold temperature. If the temperature goes up, they are going to be permeabilized and be too fluid very quickly at what for you and me would be like a moderate temperature. That's always the price, I guess. If you're so flexible and fluid that you still can move and function normally at a cold temperature, it means that as the temperature increases to moderate, you're going to denature or you're going to permeabilize, whether you're an enzyme or a membrane. So you could think about psychrophile membranes as being more like oil and less like butter. So the actual components of the membrane will be closer to like oil and less like butter. Butter is solid at room temperature. Oil is generally liquid at room temperature. They also will produce special molecules called cryoprotectants. Basically, a cryoprotectant is anything that is going to prevent freezing. So you can have some proteins that are cryoprotectants. Other cryoprotectants are not proteins. Glycerin is a good example of a cryoprotectant. Antifreeze as a cryoprotectant. Well, an antifreeze is basically also a general term for something that prevents freezing. The purpose of these cryoprotectants is to prevent the cytosol of the cell from freezing. If the cytosol freezes, then it's no longer going to be liquid. All the things that were dissolved in there, they're no longer going to be dissolved, and you're not going to have reactions occurring anymore because things cannot move through that ice. You also might have ice crystals puncturing the cell membrane. So if the cell actually freezes inside, it will die. In order to prevent themselves from freezing, even though the things around them are frozen, psychrophiles will use cryoprotectants. In this diagram here, you see some of the adaptations that psychrophiles have for the cold. They have special components in their membranes, like carotenoid pigments that prevent the membrane from getting too stiff, that make it more fluid. They also have their cryoprotectants in here. So these are both different types of proteins that act as cryoprotectants. And then they would have the special enzymes as well that are more flexible than a regular enzyme. Next we'll talk about thermophiles. Thermophiles like places that are warm, pretty hot, or that are sometimes quite hot. Most of them will live in the soil. During the day, the sun heats the soil up and it can reach a very high temperature, but then at night it cools down. So a thermophile will generally be able to tolerate like lower temperatures, not like cold temperatures, but more moderate temperatures. They can tolerate it as long as it's getting hot. Sometimes that's enough for them. You also will find them living near a hot spring. They won't live in the hot spring because the hot spring itself is too hot. They'll live nearby the hot spring in the area that's warmed by the hot spring. So this image here is a hot spring in Yellowstone Park. The color is caused by different microbes. So you have this yellow rim around it. Those are microbes that like it quite hot. They're right next to the hot spring. And then as you proceed out from there, you get into this orangey area. Those are all going to be thermophiles. They're living nearby the hot spring. They like it hot, but they don't like it crazy hot. So this hot spring, the proximity of the hot spring is maintaining a nice temperature for them, but they wouldn't be able to live right next to it or inside of it. You can also see this orange is kind of like moving away from the hot spring in waves, kind of wavy. That's because you actually have water like running off from the hot spring along these trails. As the water leaves the hot spring, it's going to cool, and so it's not as hot as it is in the actual hot spring, but it's still hot enough to make these thermophiles happy. So where you don't have water leaving the hot spring, those orange thermophiles will just stay kind of near it. Where you have water running off from the hot spring, the thermophiles can follow that water and it makes it nice and warm for them. This is also a trail of water that's coming from a hot spring that's warming up this area so that thermophiles that are here, these are green thermophiles, they're able to live here in the warm water from that hot spring. Then you also have the hyperthermophiles. They need it to be always hot. So they cannot take it. If it cools down at night, that's too cold for a hyperthermophile. It's going to die. It has to be hot all the time for a hyperthermophile. So they're going to live actually inside of the hot spring or right by a deep sea vent, places where it's always real hot. And those are their main living locations. They're usually going to grow really fast. Since they're adapted to these real high temperatures, their reactions are going to go real fast, or they can tolerate temperatures at which chemical reactions are going to proceed really quickly. It's very hard to culture a hyperthermophile because you need it to basically be like boiling or even hotter than that, depending on the exact hyperthermophile all the time in the culture. So that's very difficult to achieve. In this image here, you actually have a hot spring. This is boiling water. And if you took a sample from that, you actually have these hyperthermophiles living right in that boiling water. So they need it boiling. That's very hot. There's also some hyperthermophiles that like temperatures that are over the boiling point, which is really hard to achieve in culture. In order to get temperatures of water that are above the boiling point, you need to add pressure. So you would have to have a pressurized vessel for them, and never take them out of it, basically. So it's real hard to like study that. Of the hyperthermophiles, they are all prokaryotes, so there are no eukaryotes that can stand that type of temperature. Most of the hyperthermophiles that we know about are archaea. Thermophiles and hyperthermophiles also have adaptations that allow them to survive and grow at high temperatures. So first, their enzymes are designed for high heat, which means that they're more stable than normal enzymes or less flexible. So psychrophiles have enzymes that are more flexible than normal, and thermophiles and hyperthermophiles have enzymes that are less flexible or more stable than normal. So a less flexible membrane, or sorry, a less flexible enzyme has a harder time changing its shape. It has a harder time undergoing a conformational shift. But then you add in the greater flexibility that's caused by high temperatures, and you get an enzyme that is ideally stable and flexible and able to both maintain its folding and undergo conformational shifts for its function. So the enzymes of thermophiles and hyperthermophiles are very useful for us. There's a lot of procedures that we do in the lab or in industry that need to be carried out at a high temperature, and that we would like to have an enzyme for, right? If there's some reaction that needs to happen in this procedure, and an enzyme will make that reaction go much faster. But most enzymes will not withstand the temperatures. So you need to use an enzyme that is coming from a thermophile or a hyperthermophile so that it's able to actually function at the high temperature that this procedure would be carried out at. They also, thermophiles and hyperthermophiles will also have membranes that are especially designed for high heat, which means that they are less fluid than normal. So psychrophiles have membranes that are more fluid than normal, and then cold temperatures will stiffen them up to an ideal fluidity. Hyperthermophiles and thermophiles have membranes that are less fluid than normal, and then the heat will make them more fluid or permeabilize them partly, or go in that direction. They won't actually be permeabilized because they're less fluid than a normal membrane, and you end up with ideal fluidity for the membrane. Right, so the membrane of a thermophile or hyperthermophile is able at high temperatures to both allow substances to cross it as normal and keep things in that need to stay in, keep things out that need to stay out. So it's an effective barrier, and it works for transport. If you brought the temperature down to like what would for us would be a normal level, then the membrane of a thermophile or hyperthermophile would be too stiff for transport across the membrane, and it would die. So you could think of the membranes of these microbes as being more like butter and less like oil. So more solid at room temperature, but then when you heat it up, they become properly fluid. There's actually some archaea that have a monolayer membrane instead of a bilayer. So all the basically all the most organisms have a membrane that is a lipid bilayer, and the membranes that we've talked about so far are lipid bilayers. But for these archaea, they're generally hyperthermophiles. They'll have a monolayer membrane where the two sides of the membrane are essentially always attached to each other covalently. So here you have a bilayer membrane where you have one layer on the bottom and a separate layer on the top, and they're not actually attached to each other. In these archaea, these hyperthermophile archaea that have a monolayer membrane, the two sides are permanently attached to each other. So this membrane is much harder to permeabilize because the two layers are attached and are essentially only one layer. Another environmental variable that's important for microbial growth is pH, which is the negative log of the hydrogen ion concentration. A hydrogen ion, or H+, is a proton. Since a hydrogen is nothing but an electron and a proton, if you take away an electron to get an H+, or a hydrogen ion with a positive charge, that means that all you have left is that proton. So H+ is used to abbreviate proton. We say that pH is a negative log because when you have a lower pH value, that means you have more H plus or more protons around in the solution. If you have a higher pH value, that means that you have fewer protons in the solution. And we say that it's a log because a difference of 1 on the pH relates to a tenfold change in the amount of protons in the solution. So if you're starting at pH 7, which is neutral, and you go down to pH 6, you're actually gaining 10 times as many protons. If you go down again to pH 5, you're gaining 10 times more again, which means if you go from pH 7 down to pH 5, you're actually gaining 100 times more protons. So if you have a low pH or it's very acidic, that will tend to denature proteins. It interferes with the bonds that stabilize their structure and causes them to unfold. So different proteins will have a different range of pH values that they tolerate, but in general, when the pH gets too low, they're going to denature, which would mean that they lose their function, and if they're a critical protein, that means the microbe, depending on that protein, is going to die. pH also affects microbes by affecting the charge on amino acids and other molecules in the cell. As a general rule, if the pH is high, that means you don't have very many hydrogen ions or very many protons in the solution. Molecules that can lose a proton will probably do so. If you have a low pH and there's a lot of protons around in the solution, molecules that can gain a proton will probably do so. And anytime that happens, the charge is going to change. Especially for the acidic and basic amino acids, if you have an acidic amino acid, it's going to tend to lose a proton. If you have a basic amino acid, it's going to tend to gain a proton. Every amino acid will be affected differently, but if the change on that amino acid is if the charge on that amino acid is changing, that can affect the function of the protein. So sometimes a protein will need to have amino acids that have a negative charge or a positive charge in the active site. And also those charges on the acidic and basic amino acids can affect the stability of the protein. So this graph here is showing how a couple different amino acids are affected by pH. On the bottom you have glutamate, on the top you have lysine. So for glutamate, at a very low pH, it'll have a charge of 0. And as the pH goes up, around pH 4, it will lose a proton, and that means that it gets a negative charge. So glutamate that's over pH 6 will have a negative charge. Glutamate that's pH 3 or below will have no charge. Then for lysine, the situation is different. It's affected differently by the pH, but it's still affected. So for most pH values, lysine will have an extra proton, giving it a positive charge. Right around pH 10, it will start losing that proton, and so it ends up with a neutral charge or no charge at a very high pH. In general, every microbe, same as for temperature, every microbe is going to be adapted to the pH of its natural environment, whether that's acidic, basic, or neutral. Most natural environments are in the pH range of 3 to 9. There are some exceptions, but most natural environments would be in that range. And just like we can group microbes based on their temperature tolerance, we also group them based on their pH tolerance into 3 major groups. The acidophiles that like a low pH or an acidic environment, the neutrophiles that like a medium pH, kind of a neutral pH, and the alkylophiles that like an alkaline environment with a high pH or a basic environment with few protons around. So even if you're an acidophile or an alkalophile and you like to have a low pH or a high pH in your environment, your internal pH is probably more like a neutrophile. It's more like a neutral value. So you're going to have some type of mechanism for maintaining a more neutral pH even though the environment you're in is acidic or basic. So they'll have special mechanisms to achieve those neutral values. Which is why things will go awry if you put them into an environment that has a different pH than what they're used to. For human pathogens, most of them are going to be neutrophiles. There's a couple of exceptions to that. The majority of them are neutrophiles because your body has a neutral pH. It's about 7.4. Should be about 7.4 if it's being correctly maintained. So that means that pathogens that would make you sick are going to be happy at that neutral pH, but also your microbiome members that live inside of your gut, they're usually neurophiles that like a neutral pH. There's some exceptions to that. So your skin is actually a little bit acidic. So you can have some microbes on your skin that like it more acidic in your microbiome, and also sometimes pathogens. Your stomach is very acidic. H. pylori is a bacteria that lives inside the stomach, so that's an acidophile that's adapted to that very low pH, and it causes gastric ulcers and other problems. Also, the vagina has a low pH. It's like a moderately acidic pH, around pH 5 ideally. So the microbiome of the vagina would be having more acidophiles in it, more acidophiles in it that like that low pH, or at least are able to tolerate it. For alkalophiles, an important point or an interesting point about alkalophiles, microbes that like a high pH, is that a lot of them will not be relying on the proton motive force. Since they're living in an alkaline environment that has very few protons in it, it doesn't make sense for them to use a proton concentration gradient for their energy or for their movement, where you have a lot of protons outside the cell and a few inside. You already know, if this is an alkaline environment, that there's not a lot of protons outside the cell. Manufacturing that condition would be very difficult for them. So they'll frequently use something else. They can use different things, but a lot of them are going to use a sodium motive force instead, where they have a lot of sodium outside the cell and only a little bit inside the cell. So they can just substitute some other ion for protons. Water is also an important variable that affects microbes, and there's multiple ways that water can affect microbes. So first, you have the basic presence of water. Is it there or is it not? Most microbes need there to be at least some water in their environment. But then it's also important how many solutes are in that water and how those solutes are interacting with the water. So osmolarity is the total solute concentration per liter of water. That includes all the solutes, no matter what type of solute it is. Everything that's dissolved in the water will contribute to the osmolarity. And this is going to affect how water is moving in and out of cells by osmosis. In osmosis, water is attracted to a compartment that has more solutes, and is basically trying to even out the solute concentration on either side of the compartment. So for the cell, the compartment would be the cell. And water is going to be moving into the cell or out of the cell by osmosis, trying to equalize the solute concentration inside and outside of the cell. If you have a hypotonic solution, that means it has less solutes than the cell. If you don't have anything to protect the cell there, then you're going to have water rushing into the cell. There's more solutes in the cell, so water is always going to move to where you have more solutes to equal out that solute concentration with the outside that has few. Water will rush into the cell will swell and burst. Most microbes will prevent this from happening to them using their cell walls. So most microbes are protected from a hypotonic solution by their cell walls. But you also have sometimes a hypertonic solution where you have more solutes in the solution in the water than what you have in the cell itself. Then water is going to be leaving the cell and moving into the environment, trying to equal out the solute concentration inside and outside of the cell. If that happens, the cell is going to be losing water. If it loses too much water, it will die. So in the diagram here, you have those three kind of fates. So this is a hypotonic solution where you have more solutes, more solutes in the cell, less solutes outside the cell, and water is going to be entering the cell and swell it until maybe it pops if it doesn't have a cell wall to protect it. And here you have a hypertonic solution, more solutes outside of the cell, fewer solutes inside of the cell. Water will be leaving the cell and it will shrivel up and potentially die. For most microbes, a hypertonic solution is more dangerous than a hypotonic solution, just because microbes have cell walls specifically to protect them from a hypotonic solution. Also, though, a hypotonic solution is more common to find than a hypertonic solution. So the hypertonic solution is more dangerous, but also less common. So most microbes don't really have to worry about it. If you have high salinity, that means that you have a lot of salts or electrolytes dissolved into this water. That means that's a hypertonic solution most likely. Also, if you have very high sugar in the solution, that's hypertonic as well. If you have a lot of salts dissolved in your water, not only are you impacting the osmolarity, making it a hypertonic solution, but you're also going to be changing the water activity. And the water activity is defined as the ratio of water that's available to interact compared to the amount of water that is not available to interact with the cell, basically. So the cell can only use the water that's available to be used by the cell. Some of the water will not be available because it's already interacting with something else. For instance, a salt. If you have a salt that you dissolve in water, like NaCl, that's going to come apart into a sodium ion and a chloride ion. So all salts, when they dissolve in water, will come apart into two ions. Those ions are going to end up surrounded by a shell of water molecules that are interacting with the charge on that ion. Those water molecules are not available to interact with the cell. And you can see that in this image here. Here you have your sodium ion, here's your chloride ion, and they're surrounded with a circle of water molecules. The chloride ion is surrounded with water molecules that have the hydrogen end, which is the slightly positive end of water, pointing in towards the chloride. The sodium is arranged with a ring of water molecules that have their oxygen end, which is the negative end, pointing in towards the sodium. And it's shown as a ring, but in real life it would be a sphere, a complete ball of water molecules around each of those ions. So these water molecules are not free to enter the cell and interact with the components of the cell. They're bound up by those ions. Sugar will also bind up waters in the same way, but just not as strongly. So the water activity, if you have less water activity because you have high salt, that basically means there's less water around for the cell to use. So just because there is water, that doesn't mean that it's available for the cell. So microbes can be classified into 6 groups based on their preference for different levels of solutes or water itself. First, we have the non-halophiles, which would include like sort of the normal microbes that need low salt. They need to have low solutes in their solution. So they do not tolerate a hypertonic solution. Most human pathogens will be non-halophiles because you yourself are a non-halophile. You do not tolerate high salt. Then you also have halotolerant microbes that would like to have low salt, but they are able to tolerate some amount of salt or moderately high salt levels. You do have a few human pathogens in this group that would basically be ones that live on your skin. Your skin gets quite salty because of your sweat. So a lot of the microbes that live on your skin actually will tolerate that level of salt. The ones that are going to be pathogens that would be able to cause disease need to be able to tolerate the salt on the skin. They also need to be able to live inside the body where there's not a lot of salt, so they would be halotolerant. Then you have halophiles that need a lot of salt, and extreme halophiles that need a whole lot of salt. Both of those groups would be found usually living in the ocean, which is the saltiest, consistently salty place on the earth. Then you have osmophiles that need high solutes, not necessarily salt, but just a lot of solutes. So technically, a halophile and an extreme halophile, those are both types of osmophiles. They need specifically high salt or very high salt, whereas an osmophile is just a microbe that needs high solutes. But usually the way that we use the word osmophile is to mean a microbe that specifically needs high sugar. Not just any solute, but high sugar. Although technically it just means a microbe that needs high solutes, it's usually used to mean a microbe that needs high sugar. And then finally we have xerophiles, which are microbes that need a dry environment, so they need a place to live that does not have a lot of water in it. They would usually be living in the desert. So for these organisms that need high salt or high sugar or high solutes, they're adapted to live in some hypertonic solution. Usually the way they're going to survive in that solution without shriveling up and dying is by having a lot of solutes inside the cell, extra solutes inside the cell. So they're basically going to make sure that the amount of solutes they have inside the cell equals the amount of solutes that exist outside of the cell. That way you don't have water leaving the cell by osmosis. Then the final major environmental variable that affects microbes is oxygen. Oxygen is a special type of element because it attracts electrons very strongly. You may remember from maybe, I'm not sure if you learned it in chemistry, but we definitely go over it in general biology, that oxygen is very electronegative, which means that it attracts electrons to itself very strongly. Since it attracts electrons so strongly, that means that it's very effective as a terminal electron acceptor or a final electron acceptor. So it's very useful to use it as a terminal electron acceptor when you're making ATP using an electron transport chain. It's very easy for it to take those low energy electrons that have just gone through the electron transport chain. But the fact that it attracts electrons so strongly can make oxygen quite dangerous if it's in the wrong format. So if you have just O2, O2 is fine. That is not toxic, that's not dangerous, it's stable. But if you have some molecule that involves oxygen that doesn't have a full complement of electrons, that's very dangerous. That would mean that this molecule has a single electron somewhere. So electrons like to be in pairs. They are stable when they're in pairs. If you just have a single electron, that's quite reactive. If you have a single electron on oxygen, that's extremely reactive, since oxygen attracts electrons very strongly. And we would call that a reactive oxygen species, or ROS. Some of the main examples of ROSs would be superoxide, which is O2 minus. That's O2 that has lost an electron, so it has one unpaired electron. Also hydrogen peroxide, which you would be familiar with, H2O2, and the hydroxyl radical, which is OH with just one free electron. That can also be written as OH minus. So any of those are going to be extremely reactive. Those are all reactive oxygen species that attract electrons very strongly. Well, that basically means that they are able to strip an electron off of something else. They can strip electrons off of proteins, which would potentially make that protein non-functional. They can also strip an electron off of DNA, which would damage the DNA. And if you have enough of that damage to your DNA and to your proteins, you're probably going to die. So all organisms have a challenge with reactive oxygen species and protecting themselves from reactive oxygen species. Any organism that does respiration is going to be making reactive oxygen species as a result of that respiration. So when you are using aerobic respiration and you're using O2 as the final electron acceptor to make water, well, you're starting with O2, but water only has one O in it. It's H2O, not H2O2. That means that you need to split that O2 in half in order to use it as a final electron acceptor to make water. So usually that means that you're going to do it twice and make two waters. Each of those oxygens will go to make one water, but sometimes things go wrong. Sometimes one of those oxygens ends up making a reactive oxygen species. One of the common ones, a superoxide, hydrogen peroxide, or a hydroxyl radical. Also, some of the carriers on the electron transport chain that carry electrons around, like the quinones specifically, well, especially the quinones, but also some other ones, they are able to kind of end up generating ROS, or reactive oxygen species, when they carry oxygen. So organisms that use aerobic respiration will make ROS when oxygen is used as the terminal electron acceptor, but organisms that do anaerobic respiration, that don't use oxygen for their terminal electron acceptor, will still make some ROS because of the quinones that they use and some of the other things that they use in their electron transport chain. Even organisms that don't do respiration at all will still use quinones for different purposes. So anytime you're using a quinone or some of these other electron carriers in the electron transport chain, even if it's for a different purpose, it's still going to generate a small amount of reactive oxygen species. So organisms will be adapted to kind of tolerate that. Organisms are going to use antioxidants to react with their reactive oxygen species before those ROS can react with the proteins and DNA that make up the cell. So when you're eating antioxidants, the way that that would actually help you is by protecting you from these ROS that are generated in the process of your normal metabolism. However, some microbes have more antioxidants and some have less. So some of those antioxidants also are in the form of enzymes that are designed to react that reactive oxygen species with something else to make it safe. Some microbes have those enzymes, some microbes don't. Microbes that do aerobic respiration are always going to have a good amount of antioxidants and a good amount of enzymes that can react that react that reactive oxygen species to make it non-toxic. Some of those enzymes would be catalase, peroxidase, superoxide dismutase. You may have heard of catalase and peroxidase. When you cut your apple open and you wait and it turns brown, that's because your catalase in the apple is reacting with oxygen. Some microbes don't have a lot of those enzymes, especially if they're not doing aerobic respiration. They're not going to have a lot, or they might not have any at all. And they're going to be very vulnerable to reactive oxygen species, which means that they're not going to be able to tolerate oxygen, or not very much oxygen at least. So we can group microbes based on how they tolerate oxygen or how they use oxygen. Broadly, we categorize microbes into aerobes or anaerobes. Aerobes are microbes that do aerobic respiration. Anaerobes are microbes that do not do aerobic respiration. Some of them are doing anaerobic respiration, some of them don't do respiration at all, and they only use fermentation, but they're still going to have the quinones. That they're using them for something else, and those quinones will generate reactive oxygen species if oxygen is present. Then we have subgroups within those. So for the aerobes, we have obligate aerobes that have to have oxygen to grow. They cannot do without it. Then we have facultative aerobes that can use oxygen if oxygen is around. If there's no oxygen, they're going to do something else. Some of them will switch to fermentation. Some of them will switch to anaerobic respiration. A good example of that would be yeast, is a facultative aerobe that will use oxygen for aerobic respiration if it's present. If it's not present, it'll start using fermentation instead, making alcohol as a product, as a byproduct. Then you have microaerophiles as well. Those are not as common. A microaerophile uses aerobic respiration but doesn't have enough detoxifying enzymes to tolerate normal oxygen levels. So they live in an environment where there is a little oxygen but not a lot. If they're exposed to the air, that's too much oxygen for them, but they still need a little bit. Then for the anaerobes, we have two different groups, or two different subgroups. We have the obligate anaerobes and the aerotolerant anaerobes. The obligate anaerobes cannot tolerate oxygen at all. If they are exposed to oxygen, they will at best not grow, and at worst they will die. The aerotolerant anaerobes don't use oxygen for aerobic respiration, but they do tolerate it, which means that they do have the enzymes that they would need to detoxify the reactive oxygen species that are generated in the presence of oxygen. And we do generally a simple test to tell what group a microbe falls into, which is just basically putting media in a tube. where part of the tube is exposed to the air, or part of the tube is filled with air, and then just seeing where the microbes grow. So when you have a tube like this that's filled partly with media and the rest of it is air, the part of the media that's right by the air is going to have oxygen in it. And when you go deeper into the tube, like at the bottom, you're not going to have oxygen in there as long as you're not disturbing this tube. If you're shaking it, oxygen is going to go around the whole tube, but if you just let it sit and you don't disturb it, oxygen will be found at the top of the media, but not at the bottom. So an obligate aerobe will be growing only at the top of the tube where there is some oxygen in the media. They have to have oxygen. They cannot grow without it. An obligate anaerobe will be growing only at the bottom of the tube, and you'll see less and less of that microbe the further up the tube you go. The further up you go, the more oxygen there is. They're only going to be highly concentrated at the bottom where there's like none. A facultative aerobe will be growing throughout the whole tube, but it'll mostly be growing at the top. So this organism tolerates oxygen, also is able to live without it, so it grows throughout the whole tube, but when oxygen is present, it switches to aerobic respiration and makes more ATP that way. Therefore, you see more of that organism at the top. When there's oxygen around, they're able to make more ATP, therefore they can divide faster, therefore they accumulate at the top where you have oxygen. But if there's no O2, they're able to switch to something else like fermentation, so you do still see them growing throughout the whole tube. A microaerophile will be growing just below the area of the media where you have high oxygen. So they need some oxygen. They're not going to grow in the bottom of the tube. They need some oxygen, but they can't tolerate a lot of oxygen, so they will not be growing at the top where there's the most oxygen. They'll be growing just beneath that. Then finally, you have the aerotolerant anaerobe, which grows freely throughout the whole tube. It is using either fermentation or anaerobic respiration. It's not using oxygen at all, which means it doesn't grow faster if oxygen is there, but it does tolerate oxygen perfectly well, so it doesn't grow less if oxygen is there either. So it just grows equally throughout the whole tube. Okay, so now we'll switch to controlling microbial growth, and specifically we'll be talking about microbial growth control in the environment, not in your body, which means we're not going to talk about antibiotics. So first we've got some definitions. First off, sterilizing or sterilization means destroying all microbes. Literally every single one is dead. It's very hard to do that. Usually when you are using some antimicrobial agent, you're going to destroy some of the microbes, maybe even most of the microbes, but not all of them. Also, different microbes are harder to kill than others. Like spores, endospores, are very hard to kill. Some viruses are very hard to destroy. So to sterilize completely is very difficult. Usually we're not going to be sterilizing things completely, we'll be disinfecting things instead. When you're disinfecting, that means that you are killing microbes on a surface or on an object, and especially pathogens. So usually when we talk about disinfecting, we don't mean just killing, the halophiles that don't affect you anyways. We mean killing pathogens that could hurt you. We would be doing that using an antimicrobial agent. An antimicrobial agent is something, either chemical or physical thing, that prevents microbes from growing or causes them to die. So that could be a chemical like bleach or alcohol, but that could also be some physical thing, just like temperature or radiation or changing the pH. Anything that kills microbes is an antimicrobial agent. Some microbes are vulnerable to some antimicrobial agents, but not to others. And some microbes are resistant to a lot of different antimicrobial agents, like the endospores and some viruses. And we'll usually categorize these antimicrobial agents based on whether they actually kill microbes or just prevent them from growing. So an antimicrocidal agent, or a microcidal agent, I should say actually, a microcidal agent is something that kills microbes. A microstatic agent is something that prevents them from growing but does not kill them. And you would usually know that based on trying to grow them and failing. So if you apply a microstatic agent to some microbes, they don't grow. Then later on, you remove it and you try to grow them, they will grow because they're not dead. If you apply a microcidal agent to microbes, they don't grow. Then later on, you remove it and you try to grow them, they won't grow because they're dead. So some examples of those words, of those suffixes, would be a fungicidal agent or a fungicide. That's something that kills fungi. You could also have a bactericidal agent that kills bacteria. I've never really heard of an archaeocidal agent, but I guess you could use that word. We usually don't have a need to kill archaea since they don't cause disease. A bacteriostatic agent doesn't kill bacteria but does prevent them from growing. A fungistatic agent wouldn't kill fungi, but it would prevent them from growing. So cidal and static will be used as suffixes, and you can just add that to a word that describes whatever specific type of microbe is being affected by that agent. We measure the effectiveness of an antimicrobial agent using the decimal reduction time, which is the time it takes for 90% of microbes to die when that agent is applied. So most of them are going to be dying at first. Within the first decimal reduction time, 90% of them will die. If you want to kill all of them, you're going to have to wait for a long time. So let's say that your antimicrobial agent had a decimal reduction time of 1 minute, and you have a population of 10,000 microbes that you want to kill. So within the first minute, 90% of them will die. You're going to be left with 1,000 microbes down from 10,000. Most of them are already dead. But now, in the next minute, another 90% of the new population will die. So 90% of 1,000 is going to die now. So you're left, after the second minute, you're left with 100. In the next minute, the third minute, you're going to have another 90% of the 100 die, which means after that third minute, you still have 10 left. After the fourth minute, you would have 1 left, and you would have to wait at least 5 minutes for all of them to die. So even though most microbes will die within the first decimal reduction time, if you want to kill them all, you're going to need to apply that agent for a long time potentially, or for a decent amount of time. Depending on how many there are. Actually, if you had a population of 10,000 microbes, that wouldn't really be a lot. That would be a pretty small population for microbes. We would usually be measuring their populations in millions per mil. So the way that we're defining death also is a little funky. It doesn't just mean not alive, it means not able to reproduce anymore. Since when you see a bacterial cell under the microscope, you can't really tell, is it alive or dead? It's still a cell that's present. We are defining death of the cell as inability of that cell to divide and create a new cell. So if you've exposed your population of microbes to a bactericidal agent, the bacteria in that population will all die if you've exposed for long enough of a time. And then if you try to grow that population, you're not going to be able to grow. So the viable cell count is going to go down. The amount of microbes that are able to grow will go down, but the total cell count is going to stay constant, because those dead cells are still cells that are there, contributing to the total count. If you applied a bacteriostatic agent instead, then the viable cell count will level off while the agent is applied. Then if you remove that agent and try to grow the cells, they're going to start growing again, and the viable cell count will go up. Right, so if you use a cidal agent, a microcidal agent, the cells will actually die, which means when you try to grow them, they do not grow. If you use a microstatic agent or a static agent, then when you try to grow the microbes later, they will grow. They are prevented from growing only as long as that agent is applied, and then they grow after. There's a couple of other terms that we can also use to measure the effectiveness of chemical antimicrobial agents: the minimum inhibitory concentration, or MIC, and the zone of inhibition. Every chemical antimicrobial agent has a MIC, which is the lowest concentration of that agent that you can apply to microbes, and it will prevent all of their growth. Not just most of their growth, but all of their growth. Then the zone of inhibition is a more specialized term. So when you're determining the zone of inhibition, you're going to take a petri dish and plate it with microbes. You're going to spread the sample of microbes evenly across the entire petri dish. Then you'll take a little circle of paper and soak it in the chemical antimicrobial agent that you're trying to measure. And you're going to place that piece of paper onto the petri dish. Then you incubate the cells and let them grow. They're going to end up growing anywhere that you have less than the MIC concentration of that antimicrobial agent that you used. So for most of the plate, it's not going to have any of that chemical antimicrobial agent. The cells will grow just fine. Right by the paper, though, you're going to have some of that chemical agent diffusing off of that paper and into the medium around it. And you're not going to have microbes growing there. So the zone of inhibition is the size of that zone around the piece of paper where you do not see microbes growing. If you have a very effective chemical antimicrobial agent, you're going to have a very large zone of inhibition. If you have a not very effective chemical antimicrobial agent, you'll see a small zone of inhibition. This is really useful for testing the susceptibility of a specific type of microbe to different chemical agents. You could also be using proper antibiotics as well that would be intended for internal use in a human. In this type of test, you would use multiple pieces of paper that are all soaked in a different compound that is some antimicrobial agent, and you're going to see basically where do the microbes grow and where do they not grow, or which one of these compounds has the biggest zone of inhibition and which one has the smallest. So that's the type of test that's being shown in this diagram, where you have your sample of your microbes and you plate them evenly on this petri dish, and then you put some pieces of paper. Each piece of paper is soaked in a different antimicrobial agent, maybe a different antibiotic. Then you incubate and you see the zones of inhibition. It's going to be a clear area around each circle of different sizes. For this particular microbe that was tested, this antimicrobial agent on the purple paper actually does not affect those microbes at all. So there is no zone of inhibition around that one. So if you have a microbe and you think it may be resistant to certain antibiotics, you might put different antibiotics on these papers and test it that way. If you see a big zone of inhibition around the paper for one antibiotic, that's a very effective antibiotic. If you see no zone of inhibition around an antibiotic, that means that that microbe is resistant to that antibiotic. So now we'll go through some of the most common physical antimicrobial agents that are used. The first is heat, which can be used to disinfect or to sterilize. Moist heat will always work better than dry heat to sterilize or disinfect things. This is partly because air insulates. So if heat is being transmitted through the air, then it doesn't transmit as well as it would through a liquid medium. So we usually like to use moist heat to disinfect or sterilize things by heat, but there's some things that can't be exposed to moisture, like special pieces of equipment that can't be exposed to moisture. Those things could be exposed to dry heat using an oven or even an open flame. When you're doing that, you have to expose the equipment to the heat for longer than you would if you were using moist heat, because it takes longer for the heat to transmit through air than it does through liquid. But the most common way that you would use heat to sterilize things is the autoclave or autoclaving things. When you autoclave something, you put it in a special machine that creates heat under pressure. You would always have your item sitting in a tray of water, and that water is going to end up boiling. But since you have pressure generated by the autoclave, you can achieve a higher temperature than just the boiling temperature of water. So if you just boil water, it's never going to go over 100 degrees Celsius or 200 degrees Fahrenheit. 212 degrees Fahrenheit. It's never going to go over the boiling point of water. The boiling point of water does vary a little bit depending on your elevation due to air pressure, but basically 100 degrees C, 212 degrees Fahrenheit is the maximum you can achieve with just boiling some water. But there's some microbes that are not killed at those temperatures. So if you want to sterilize stuff, you need a higher temperature than the boiling point of water. And you can only achieve that if you use pressure. Water that's under pressure will boil at a higher temperature than the boiling point. So autoclaving is really effective. It'll basically sterilize things. There's only one exception, which is the prion, which you may have heard mentioned in a biology class before, or perhaps not. A prion is not a microbe. A prion is not alive. A prion is a protein that is misfolded that causes nearby proteins of the same type to also misfold the same way. Prions are responsible for several diseases, the most famous of which is mad cow disease. The diseases caused by prions generally would affect the brain, and they're neurodegenerative diseases where you have it for many decades, and it's just slowly damaging your brain for that time period, and eventually it kills you. The prion protein is extremely stable. It's extremely hard to denature it, and autoclaving will not do it. So that's not generally relevant, but if someone is known to have a prion disease, then just autoclaving equipment that you used on that person wouldn't be enough. The prions will survive the autoclave. They don't really survive it because they're not alive, but they'll maintain their infectiousness and their pathogenicity despite autoclaving. Everything else will be dead or destroyed. So autoclaving is what we would generally use in the hospital or in the lab to sterilize anything that can withstand the autoclave. Any equipment that can withstand autoclaving would be autoclaved to sterilize it. A lot of the time in industry, they'll use something else, a process called pasteurizing or pasteurization. In pasteurization, you are heating something, but not enough to kill all of the microbes. You're heating it enough to kill many of the microbes, or even most of the microbes, but not all of them. And you're specifically not heating it so much that you damage the product. So a lot of beverages that you buy will say that they're pasteurized on it, like milk or juices, which means that they heated it, but not so much that they destroyed the milk. But enough to kill most of the microbes in there so that it's not sterile, but it'll last for a lot longer that way. If you burn something, by the way, it will be sterilized perfectly, completely. Even prions will be denatured and gone, but a lot of things will be damaged by burning them, right? Most equipment, you can't just burn it and expect it to work the same afterwards. So that's generally not used. Autoclaving would be used instead. Refrigeration is microstatic for most microbes. So the purpose of your fridge is not to kill microbes, but it's to keep them from growing by having a temperature that's too low for them. There are some microbes that still grow in the fridge, like cycrotolerant microbes will still grow in the fridge, like the Listeria monocytogenes will grow in the fridge okay. But for most microbes, refrigeration is microstatic. You can also freeze microbes. But a lot of microbes actually won't die when you freeze them. Scientists will also use freezing to preserve samples of microbes for the future. When they're doing that, they will actually immerse the cells in cryoprotectant so they don't actually freeze. That way they're still basically normal and viable when you thaw them out and try to work with them. So freezing is not generally used to disinfect or to sterilize things, but it is used very commonly to store samples with cryoprotectant to prevent the actual cells from freezing. Temperature will also affect the decimal reduction time. Some organisms, of course, survive better at a different temperature than others. So moderate heat might be deadly for a psychrophile, but it would not be deadly at all for a thermophile. So when you're considering the decimal reduction time of temperature, it doesn't have like a standard decimal reduction time. It depends on the specific microbe that you're trying to kill using temperature. Also, for a chemical agent, the effectiveness of that agent might be affected by temperature, and they're usually more effective at a higher temperature. So the decimal reduction time usually goes down as temperature goes up. Next we have radiation. Usually we would use UV light to sterilize things. UV light can interact with DNA and be absorbed by DNA. When that happens, the DNA is damaged, which may lead to a mutation, or it may just lead to a break in the DNA. Not all UV light actually will be absorbed by DNA. Only UV light in the specific range of 220 to 300 nanometers of its wavelength will be absorbed by DNA. That type of UV light is dangerous for all organisms that use DNA. So all microbes will be damaged by UV light and will be killed by it. So if you just have a little bit of UV light that the microbe is exposed to, maybe only some of its DNA is damaged and it's not that bad, and it can actually repair the damage and survive. But if you bombard the microbe with consistent UV light, it's going to die. The drawback of UV light is that it doesn't go through objects. So you have to have a surface that is actually exposed to the UV light for it to be effective, which means that if you're going to use UV light to sterilize something, you have to make sure that it is completely clean beforehand. With like anywhere you have a shadow, the UV light will not be sterilizing or killing anything in that area. So UV light is commonly used to sterilize hoods where we work with microbes in the lab. Before you do that, you have to clean that whole hood out, and then you shine the UV light and everything in there dies. There is going to be actually an exception. There's this little hose here, which would be like maybe a vacuum line hookup or a gas line hookup. Underneath that hose, you have a shadow there. You don't have UV light actually hitting that surface, so that part will not be sterilized by this UV light. So the fact that you have to completely scrupulously clean something and eliminate all shadows kind of limits the effectiveness of UV light. You can also use ionizing radiation, which is radiation that has enough energy to take electrons off of atoms, which would make ions, which is why it's called ionizing. So up here I have the electromagnetic spectrum. Here you have your ultraviolet light. Here's the visible light, then ultraviolet light, then X-rays, then gamma rays. As you go down on the spectrum, the wavelength is going down. It's getting shorter and shorter. When you have a shorter wavelength, you're going to have more energy in these photons. So ultraviolet light has a good amount of energy, but it is not ionizing. It does not have enough energy to strip an electron away from an atom. If you continue going down into X-rays and gamma rays, the energy is increasing as the wavelength decreases, and you end up with enough energy to create ions by taking away electrons. This is very bad for any biological molecule that is exposed to X-rays or gamma rays. It'll damage proteins, damage DNA. You can tolerate a small amount of it, but too much will kill you. The benefit of ionizing radiation is that it does penetrate objects. So you don't have to be as careful about eliminating like all shadows or just making sure that every surface is exposed to the light. But the bad thing about it is that it's more dangerous for you, and since it can penetrate objects, it's more difficult to kind of use it but not expose yourself to it. Finally, you could use filter sterilization, which doesn't really kill anything, but it just takes things out of the fluid that you're working with. So you can filter any fluid, and fluids technically include liquids and gases. So that means that you could filter air and you could filter media, any liquid or gas. You're not actually killing the microbes, but you are removing them from that liquid or that gas. So when it comes out the other side of the filter, it doesn't have microbes in it, and you can consider it sterilized. This would be useful for fluids that are damaged by heat or by radiation. You could filter them instead. Usually filtration doesn't actually sterilize things. How effective it is going to depend on the pore size of your filter. For sterilization, you have to use a pore size that is 0.2 micrometers or smaller, no bigger than that. But if you're using a 0.2 micrometer pore, most viruses will still be able to get through that. So if you had viruses in that liquid, it would not come out sterile on the other side. It would come out disinfected with bacteria removed, and cellular microbes removed, but with viruses still in it. In order to get viruses out, you have to use an extremely small pore size. The trouble with that is that it gets clogged extra easy, so it's just a little bit impractical. Finally, we'll briefly talk about the chemical antimicrobial agents. We're not going to mention specific ones, but we'll talk about different categories that they fall into. So first you have the sterilants, which sterilize, destroy literally every microbe that's present. Then you have disinfectants, which would be more common than sterilants. Disinfectants are antimicrobial agents that kill most things, but not all of the things. So some microbes that are resistant, like some viruses or endospores, would survive a disinfectant. And disinfectants are going to be used specifically on surfaces or objects, not on a person or a living thing. So a good example of a disinfectant would be like alcohol or bleach or Lysol. Those aren't going to kill everything, but they'll kill most things. They'll kill some viruses, but they won't kill all viruses. I should say destroy some viruses, but not all viruses. Then you have sanitizers, which are similar to disinfectants, but they're not as strong, so they don't kill as many things. They'll leave a larger population of microbes behind, and also more microbes will be resistant to sanitizers than to disinfectants. And finally, you have antiseptics, which are also called germicides. Those are specifically antimicrobial agents that are not toxic when used correctly for you, which means that you can use it on your own tissue. So hand sanitizer, that's actually an antiseptic. Alcohol would fall into multiple of these categories, actually. Alcohol is non-toxic for your tissues when you use it properly, so it's an antiseptic or a germicide, but it's also useful as a sanitizer or as a disinfectant. Actually, not generally as a sterilant. It doesn't kill enough things. There's a lot of factors that could affect the antimicrobial agent that you're using. Temperature, of course, will affect it. Usually, antimicrobial agents will work better at a higher temperature, but not always. It depends on the specific agent. Some agents would be destroyed by heat. It'll also depend on the pH. Some agents will work good at a neutral pH, some work better at a basic pH, some work better at an acidic pH. The time you apply it for matters a lot. So when you are disinfecting something, you have to make sure that you have read how long you need to leave that agent on there for it to actually be effective. If you just put it on and take it right off, it didn't kill what you thought it killed. And finally, the presence of other molecules, and specifically organic molecules, can affect chemical antimicrobials. A lot of those antimicrobials will be able to react with organic compounds, and when they react with those compounds, they lose their effectiveness. So in general, you want to make sure that you have cleaned the surface off before you apply a disinfectant to it for maximum effectiveness. When we're talking about organics, that means like on your counter, like crumbs. That also means dust in your home. Dust mostly comes from skin, so it has a lot of organic compounds in it. And it means biofilms too. Biofilms are very good at keeping antimicrobials out so that the microbes are not exposed to them. So if you're going to destroy microbes that are living in a biofilm, you have to physically disrupt that biofilm. You have to scrub it away. And then you can apply the disinfectant to kill whichever microbes were left behind clinging to the surface. These organics are always going to be forming a type of barrier between the disinfectant and the microbe. Right, so if you have a dusty surface, you could have microbes underneath the layer of dust and they're kind of hiding in there. Biofilms will work that way. Yeah, and they can frequently react with the disinfectant.